For decades, biology has relied on a straightforward assumption. Human development and heredity are driven by fixed, unchanging genes. We've treated the genome as a hard-coded blueprint, where precise sequence tokens lock into place to build a functional organism. This perspective was cemented by our primary human reference genome, GRCH38. It presented human genetics as a single, static, linear text to be read from start to finish. But that linear text was an incomplete map. When the T2T consortium released the CHM13 gapless assembly, the static reference was forced to expand, integrating 238 megabases of new, complex data into a structure we once treated as complete. By resolving these massive blind spots, the new assembly uncovered over 200 megabases of previously hidden sequence. Instead of simple text, researchers found dense, overlapping layers of genetic architecture that the old linear map entirely missed. The updated assembly also actively deleted established knowledge. Entire sections of the old reference were exposed as false duplications. Genes we had cataloged and studied turned out to be cartographic errors. While some genes vanished, others suddenly multiplied. The FRG1 gene family, for instance, expanded from 9 known copies to 23 newly resolved paralogs that had been entirely invisible. If a supposed genetic invariant can appear, disappear, or duplicate simply because we improved our sequencing technology, then the isolated gene token is highly dependent on the quality of the map. The fixed gene was an illusion born of an incomplete map. Our previous reference manufactured false simplicity, hiding the highly fluid reality of the genome. That fluidity becomes even more obvious when we move past a single reference genome. Recent pangenome data, sequenced from 65 diverse individuals, allows us to track variation across different human populations. The scale of structural variation is immense. Compared to the baseline reference, the average healthy individual carries over 26,000 structural variants. These variants include massive copy number shifts, large structural deletions, and significant sequence insertions distributed across the entire genome. Within a single person, hundreds of core protein coding sequences are actively disrupted by these breakpoints. Yet, despite these disruptions, such as the deletion of repeat units overlapping exon sequences and the MUC5B gene, the coding and splice structures remain perfectly functional. We are left with human genomes that have wildly different gene counts and structural architectures, which nonetheless produce indistinguishable, viable human phenotypes. This creates an unavoidable tension. If genomic sequence tokens are constantly shifting, duplicating, and breaking, how does biological form remain so rigidly organized and functional? To resolve this tension, we have to look beyond the physical sequence string and examine the higher-order organizational forces that constrain it. Think of this as a biological state space, a phase space where the raw genome sequence, regulatory topology, epigenetic domains, and physical traits all interact simultaneously. To account for stability we observe, the TCGS-SEQUENTION framework proposes a model of corridor-preserving functional organization. In this conceptual view, dozens of differently sequenced DNA strands settle into a single, unified functional corridor at the bottom. This corridor establishes an equivalence class. Multiple, entirely different genomic sequences can funnel into the exact same viable biological outcome. The biological system tolerates local sequence mutations and structural variations perfectly, as long as the overall state remains within the boundaries of this protective functional corridor. Catastrophic disease occurs when a perturbation carries enough force to push the system outside that boundary. This upends the old linear model, which assumed that any change to the sequence inevitably meant a loss of function. Biological organization persists not by freezing the local sequence in place, but by strictly enforcing the topological and epigenetic boundary of the corridor itself. We can see this corridor theory operating in hard genomic data by examining the short arms of the acrocentric chromosomes. The non-ribosomal regions of these arms are extremely volatile. They are so non-unique that they align at 98.7% identity across completely different chromosomes. This extreme similarity drives constant ectopic recombination, meaning these chromosomes are relentlessly swapping sequence material. But embedded directly within these volatile arms is a stark contrast: the ribosomal DNA, or rDNA, arrays. Despite the surrounding chaos, the rDNA arrays maintain a strict canonical head-to-tail organization. Researchers found 0 observed inversions within these structures. The arrays preserve their expected transcriptional arrangement and remain highly homogenized, completely ignoring all the sequence-level noise erupting around them. The true biological invariant is not the local sequence token, which can be swapped at will. It is the array-level architectural law that protects the system's functional geometry. The centromere provides another test of this principle. It serves as the critical mechanical anchor point required for cell division. Within human centromeres, the alpha satellite repeat arrays vary wildly in sequence length. Across populations, these sequences frequently mutate and even bifurcate into entirely new arrangements. Within that chaotic array lies the centromere dip legion, or CDR. This is a specific hypomethylated zone that dictates exactly where the kinetochore must attach. Even when the underlying array splits or reorganizes, the CDR reliably anchors itself to highly identical domains, preserving its necessary function. This protected functional domain actively repels interference. Nearly all mobile element insertions are forced to land strictly outside the bounds of the CDR. The raw genetic sequence remains highly vulnerable, but the epigenetic domain remains absolutely secure. If exact sequence identity were the true invariant, centromere mutations would cause cell division to fail. The centromere survives because the cell stabilizes the kinetochore domain, rendering underlying sequence noise functionally irrelevant. To map the true architecture of robustness, we have to abandon raw sequence conservation as our ultimate metric for biological importance. A far more accurate metric is admissibility conservation, governed by a new 4-tiered ontology of biological invariance. At the top is the shadow signature, the observable layer where traditional genes and transcripts live. They act as fluctuating indicators. Beneath that lie the domain and corridor invariants. These stable regulatory networks and epigenetic anchors dictate what is functionally allowed to exist. This structure explains why, in highly duplicated areas like the FRG1 gene family, the broader architectural layout and dosage compatibility predict biological function far better than the presence of any single canonical gene copy. A deeply conserved gene is not the source of the system's stability. It is simply a high-fidelity trace left behind by a protected functional corridor. The gene is an observable map inscription that occasionally points us toward a much deeper topological law. With this tiered framework in place, we have to rethink how we interpret genetic mutations, especially large structural variants. A structural variant operates as a topological perturbation, a kinetic impact that actively tests the boundaries of a functional corridor. The cell tolerates these perturbations differentially. A variant's biological significance is determined entirely by whether it stays within the functional equivalence class or if it carries enough force to breach the corridor boundary. A mutation can cause massive token-level damage, deleting entire genes or expanding sequence repeats while leaving the protected functional domain untouched. These findings suggest a different path for systems biology, moving past the search for immortal sequence tokens and beginning to map the boundaries of these functional equivalence classes. The map is not the territory. Our genomes exhibit a layer of structural complexity and functional resilience that exceeds the limits of a linear reference. Human biology relies on a beautifully constrained, highly robust manifold of admissible organization to survive relentless sequence edits.